DNA. Okay, so that technology is called the recombinant DNA technology, right? So usually recombinant DNA technology, we also call it as a gene cloning. Okay, we also call it as a gene cloning. So it contains a lot of experiments, a lot of protocols, okay, that will lead to the transfer of the genetic information from one organism to the other organism. Got it? So that is the recombinant DNA technology. Got it? What is recombinant DNA technology? It's a type of gene cloning, okay, so which contains a lot of experiments and protocols leading to the transfer of genetic information from one organism to the another organism. Okay. So here in the recombinant DNA technology, first DNA molecules, I mean the DNA molecules, okay, that are extracted from different sources and chemically joined together. For example, you take an animal gene, okay, then combine it with the bacterium DNA and see the expression. Okay, what the bacteria is releasing, what the bacteria is expressing based on the information present in the incorporated gene. Okay, so this recombination of DNA, it occurs naturally in microorganisms. Okay, this recombination usually it occurs naturally in the microorganism. That's the reason you can find a lot of mutant microorganisms. Okay, so in 1970s and 80s, scientists developed this artificial techniques for making recombinant DNA. Clear? In 1970s and 80s, scientists developed this artificial techniques for making the recombinant DNA. So we call this as our DNA technology, recombinant DNA technology. So what is the goal of recombinant DNA technology? So to isolate and characterize the gene, it is nothing but cutting a gene. Okay, cutting a gene. We have a big gene sequence. Okay, so just cut a part of the gene, isolate it, and then characterize what are the what is the code in that gene, what it is going to express. So that thing you can do using this recombinant DNA technology. Then to make desired alterations in one or more isolated genes, okay, you can make some desired alterations also in the genes. Okay, then it is simple. We have to cut a part from the gene and combine it with some other DNA material, okay, and place it into a bacterial cell or any other cell. Okay, that is a recombinant DNA technology. You are combining the DNA, the desired part of the DNA with other organisms' DNA. Then the other organism will express the content of the DNA. Then you will understand like what it is doing. Okay. Then to return altered genes to living cells, artificially synthesize new gene, alternating the genome of an organism. You can alternate the genome of the organism also by inserting a new gene, right? That is a recombination. So, understanding the hereditary disease and their cure also, then improving human genome by genome project. There's a human genome project, right? So you can improve that human genome also using this rDNA technology. Okay. Now, recombinant DNA technology, this is a protocol. First, we should isolate the desired gene. We have to isolate the desired gene. Okay. Then later, isolation of the vector. We need a vector. What do you mean by vector? Vector is nothing but an intermediate person, or we can call it a carrier, right? Carrier. See, mosquitoes are vectors because they carry diseases, right? They are the carriers of the disease. Okay. So flies are the vectors. Okay, vectors means like intermediate hosts, right? So isolation of the desired gene. So first you isolate a desired gene and then isolate a vector, then formation of the chimeric DNA. What is the chimeric DNA? Insert the desired gene into a vector gene. Okay, now it contains the hybrid gene. Means it is having both the vector gene as well as the desired gene. That's called the chimeric DNA. So put this chimeric DNA into a cell. Okay, maybe a human cell, maybe a yeast cell, or maybe any cell. Okay, maybe a microbial cell. Okay, any cell. You just keep the recombinant DNA. Recombinant DNA means the DNA of the desired part as well as the DNA of the vector. Okay, then put it into a cell. Okay, so what is that DNA called? Chimeric DNA or recombinant DNA. Okay, so how to put into the cell? We have different type of the methods. So here I just mentioned the transfection. There are so many methods like conjugation, transduction, transfection. Okay, like that we have different methods. So using the transfection, we are uploading or we are inserting this chimeric DNA into a cell. Okay, then Later you select the cells, grow the cells. Okay. So what are the cells possessing this chimeric DNA? You separate them because not every cell will allow this chimeric DNA, right? So some cells may allow this chimeric DNA. So isolate the cells which are having the chimeric DNA. Then express, grow them separately and see what is the product it is expressing. Okay. Then based on that, we can say that this organism is expressing the characteristics of the gene or whatever code present in the gene. Got it? So in that way, you can get the desired product from this cell. Okay, by inserting a recombinant DNA. Okay, let us see this diagram. You'll understand. First donor, the green color content is a donor DNA. Okay, so donor DNA contains a small part of the DNA, right? So what is the DNA? That's called a desired DNA piece. See, for example, okay, let us assume how the insulin is prepared, how the insulin, you know about the insulin, right? It's a normal example. What is an insulin? Those who are suffering from diabetes, usually lack of insulin production or lack of, proper insulin production may lead to the diabetes, right? So as there is no insulin in their body, it can't control the glucose. That is the reason we have to take the insulin from outside. Okay. Maybe you think about the type 1 diabetes. When it is a type 1 diabetes, then usually the people from their childhood only, they will be taking the insulin as there is no production of insulin in their body. Okay. So how this insulin is prepared? Anyway, we purchase the insulin from medical shops or from any surgical shops, right? Or medical shops. So how this insulin is synthesized, how this insulin is prepared means by rDNA technology only. How they prepare this insulin? See, there is a donor DNA. Okay, so this DNA extracted from the islet cells of Langerhans. Well, let us assume. So in that cells, this part of the DNA is producing the insulin. Okay, let us assume that only this part will produce the insulin. So this is a desired piece which produces the insulin. So cut that part, cut that part. How to cut that part? There are some enzymes called the restriction endonucleases. Restriction endonucleases means these are enzymes only, right? These enzymes cut the nuclear material or DNA. That is the reason we call them as the endonucleases. Endonucleases means it will cut the DNA in center part, not at the ends. Okay, not at the end of the DNA. If it cuts the end part of the DNA, we call it as the exonucleases. Okay, so here we are talking about the endonucleases because it is cutting the desired part of the DNA. And why we are calling it as restriction endonucleases means this enzyme will be cutting only that particular restricted site only. Got it? It won't cut the other parts. Okay. So to cut DNA in one place, then some restricted enzyme should be used to cut only that sequence. Got it? So already we know the insulin secretion DNA or insulin secretion gene code already we know. The codon already we know. Okay. Then take the islet of Langerhans cells, extract the DNA, and check that area, okay, where that particular DNA code will generate the insulin. Cut that part using the restriction endonucleases, right? After cutting, then take one more plasmid DNA. What do you mean by plasmid? Plasmid? Any idea? If you have any idea, you can just type in the window. Otherwise, leave it. What is a plasmid? Plasmid means it's an extra chromosomal nuclear material present in the bacterial cell or E. coli also. What is a plasmid? Plasmid. It's not plasma, plasmid. It's a extra nuclear, extra nuclear or extra chromosomal DNA. Means this is an additional DNA present in the bacterial cell. Okay. So we take this one, plasmid DNA. Okay, from a bacteria. Take the plasmid DNA and again cut with the restricted endonucleases. Okay, remove that place, remove some part of the DNA. Then later insert the desired DNA piece into the cut plasmid DNA. Okay, like this. And later use the enzyme called the ligase. Then it will get ligase. Ligase is for the attachment. Now what is this called? Recombinant DNA. Okay, so usually in the molecular laboratories, we do all these methods every day to find out the cancers and to produce the artificial gene. We do because we purchase the plasmid DNAs, we purchase the restriction endonucleases, we purchase the templates also. All these are available in the market. Okay, so when you work in a molecular diagnostic laboratory, or if you get a chance to, visit a CDFD, right, Center for DNA Diagnostics and Fingerprinting, it's in Nagol, nearby to metro station. So they do all these techniques, right? So if you get a time to visit there, just go there, okay, and visit there, and you can find out all these molecular biology techniques there, okay. Because I think I was there for a week, okay. Then I observed all these techniques, okay. Later we implemented the same techniques in isolation of different genes related to the cancer. Okay, we implemented all these techniques in the MNG Cancer Hospital, okay, like 6 7 years back only. Fine. Okay. So you are understanding, right? So what is the recombinant DNA? The desired piece of this, the gene, as well as a cut plasmid DNA. You keep the desired DNA piece into the plasmid. Then it will become a recombinant DNA. So connect it with the ligase or join it with the ligase. Okay. Ligase acts as a gum. Okay. Now this is a recombinant DNA. So keep this recombinant DNA in a bacteria or any cell. Okay. So here, what is the vector? This is the vector. Plasmid DNA is the vector, and this is the desired DNA. Right? Plasmid DNA is the vector. Now this is the recombinant DNA. This recombinant DNA you insert into a cell, maybe a bacterial cell, maybe a yeast cell, maybe a E. coli cell, any cell of your interest. You can place it into the human cell also. Okay, I will explain like why we place this in the human cells. Okay, so for example, uh, the human, okay, is unable to deliver or unable to produce some type of the protein. Okay, the human is unable to produce some type of the protein, let us assume. Okay, then if you insert this into a human cell, then it will express the protein, right, which the human is unable to produce. Got it? So this is how this recombinant DNA is placed into a cell. Then see, this is the actual cellular nucleus, and this is the extra chromosomal nucleus, right? That is the reason we inserted the plasmid. Okay, what's the plasmid? Which contains the DNA. Then multiplication occurs. Okay, when the multiplication occurs, it's like a cloning. All the cells, they carry the plasmid and also the recombinant DNA. Then later, based on this, the desired DNA piece, all the cells will excrete the, or all the cells will express the insulin, right? Then that insulin is collected from this cell, okay, and they will purify it, and later they will be supplied. So this is how the insulin is created, or the insulin is manufactured in a like huge content. Got it? Then what are the tools? I mean, this is just an example. Okay, so tools of recombinant DNA technology. What tools we need? Enzymes first. Why enzymes? To cut the particular part, we need to, we need to have the enzymes, right? So enzymes we need. So what enzymes? Just now I told you, exonucleases, endonucleases, restriction endonucleases. Okay, to cut in that particular part. Then vectors. What do you mean by vector? Vector means the plasmid DNA is a vector. Host. Host means you have to insert into one cell, right? Maybe a cell or a bacterial cell. That is a host. Then DNA to be cloned. Okay. Then what type of DNA you want to clone? That's a desired DNA unit, right? So desired DNA also. These are all the four things we need to, uh, we require to apply in the RDNA technology. Then these are the enzymes just now we discussed. Nucleases, exonucleases, endonucleases, and restriction endonucleases, right? Then coming to the restriction endonucleases, there is a separate nomenclature for the restriction endonucleases. Okay, let us see. Bacterial enzymes that cut DNA molecules only at restriction sites. That is the reason we call it the restriction endonucleases. Okay, so bacterial origin equal to enzymes that cleavage foreign DNA. Okay, so most of the restriction enzymes are the bacterial origin only. Okay, bacterial origin only. So the restriction endonucleases which are available in the market are mostly bacterial origin only. So they cleave the foreign DNA of desired part. Okay, so these are named after the organism. See, for example, a restriction endonuclease ECOR1 is available in the market. Okay, so this is a nomenclature. Why we call it the ECOR1? ECO means it's a Escherichia coli. Okay, E is Escherichia, CO is coli. Then R, it is a strain. R is a strain. Okay, what is the strain? RY13 is a strain. And first endonucleases. So we have different endonucleases like endonuclease 1, 2, like that. Okay, so when we purchase this ECO R1, the meaning is it is extracted from a E. coli strain, that is RY13, and that is the first endonuclease, like this. Clear? The same way, these endonucleases are two types, type 1, type 2. Type 1 cuts the DNA on both strands. Okay. Type 2 cuts both strands within the particular sequence. Type 1 cuts at a non-specific location at varying distances from the particular sequence that is recognized with the restriction enzyme. Got it? So it's a particular sequence it will cut, then it will cut at a non-specific location. Okay. Now this is the action. So this is a DNA. This restriction endonuclease is cutting in this place. Okay. What is the enzyme here? That is RESM. Okay, RESMA1. See, RE means restriction endonuclease, SMA1 or HPA1. So this is a nomenclature. Based on this, we can understand like from where this enzyme is synthesized or where this enzyme is extracted. Okay, so the action of this restriction endonuclease is cutting in this place. It cuts the double-stranded DNA like this. Later we have two blunt ends. Okay, this is action of one restriction endonuclease. Now let us take one more restriction endonuclease, that is ECO R1 and BAMHI. Okay, so what it is doing, it is cutting, the sticky ends. See, the cut started here, G. Okay, G. Okay, then it cuts like here, G, and then here, G. It cuts at G only from two strands. So the cutting happens here and then here. Now you can see this part only up to this part, and later this is a separate part. And here up to the G it cuts here. So it will be like this. These are called sticky ends. Sticky ends. Okay, I will show you later. Now these are some different enzymes available in the market. See, ECO R1. It cuts in this sequence only. Now just see G and G place, right? See, G and G. It cuts only this particular part. Then BAMHI. It also cuts in the same one. Okay, here the G is there. Then He1113, Haemophilus aegypticus. Then Hind3, Haemophilus influenzae. Then NOT, Nocardia otitis. Okay, like this. So these are the products. This is the recognition sequence, and these are the products. Okay, after cutting, you will get these type of the products. So these are the some restriction enzymes. Clear? Now, not only these restriction enzymes, we are talking about the exonuclease and endonuclease also, right? So we have different types of exonuclease, endonuclease based on the application. See, for example, alkaline phosphatase, it will remove the phosphate group. When you are doing the rDNA technology, so it is just like a tailoring work. Okay, so whatever you need, you just cut it with the scissor. Scissors are nothing but the enzymes. Okay, so cut it in different locations, and you have to attach, okay, ligase, or you have to excise one, what we say, it is just like stitching a cloth. Okay, what are all the arrangements you want to do, you have to do. Okay, remove one nucleotide, add one nucleotide. Okay, cut in that particular place, then insert this in that particular place. Then if it is missing, then you have to synthesize by adding some oligonucleotides. Okay, like that. So you have to increase the size, you have to decrease the size, you have to cut it, you have to ligate it. Okay, so all these things should happen in the rDNA technology, right? That is the reason we use different types of the enzymes. We use the alkaline phosphatase, then BAL31 nuclease, DNA polymerase 1. You know this. Already we discussed the DNA polymerase 1. Okay, DNA polymerase, DNase 1, exonuclease 3, then what do you say, restriction enzymes, reverse transcriptase, RNase A, Taq polymerase, okay, terminal transferase. Like this, we use different types of the enzymes. okay, based on the requirement. Then coming to the ligases. Just now I told you what is the ligase. What the ligase do? Ligase will ligase will join the desired DNA piece with the plasmid or the vector DNA. That's the extra chromosomal DNA. That is a ligase. Ligase also is an enzyme only, right? So DNA ligases. The cut DNA fragments are covalently joined together with the DNA ligases. So these DNA ligases also originally isolated from viruses and also from the bacterial eukaryotic cells also. These actively participate in cellular DNA repair process. Clear? So anyway, it will just ligase the phosphodiester bonds only, right? Whenever there is a cut in the phosphodiester bond, then they will ligase. It is nothing but they seal the DNA fragments, right? So these are the ligages. Later coming to the vector. So we are discussing one by one in detail. Okay. So you understood the process, right? So this is the process. Now we are discussing in detail about what type of DNA is extracted, and we are talking about the vectors now. Already we discussed about the nucleases or restriction nucleus and what are the enzymes required in the RDNA technology. Now we are talking about the vectors. Then after vectors, we will also discuss about what type of cell is needed to insert. Then later, what are the methods to insert the recombinant DNA in this cell? And later, what type of medium is needed to generate these or multiply these? And later, which one is the transfected one? Means which cell is already having the recombinant DNA and which cell is rejecting. Then we have to select the recombinant organisms and non-recombinant organisms. Then after selecting, then how to express the product. Okay. So these things we are discussing in detail. Okay. Now coming to the vectors. So we have different types of vectors. What are the vectors here? Vectors are nothing but where we are inserting the desired DNA, okay, into a vector DNA, right? So vectors are of different types. Plasmids. Plasmids are extra chromosomal uh, genetic material present in most of the microorganisms. Then bacteriophages. What is the bacteriophage? Bacteriophage means a virus that infects the bacteria. Virus not only infects the human beings, right? Even virus can infect the bacteria also. Got it? So those are called the bacteriophages. What is a bacteriophage? A virus which infects the bacteria, right? That is called the bacteriophage. Then we can use the bacteriophage also as a vectors. Then cosmids are also there. Then artificial chromosome vectors are there. Then expression vectors are there. Like this, we have different types of the vectors. Okay. Now, once the desired fragments of DNA are obtained, they are inserted into a suitable vector to produce indifferent number of copies of genes, right? This is known as gene cloning. So an ideal cloning vector should have the following properties. It must have low molecular weight. Okay. The vector should have low molecular weight. Then it must have a unique cleavage site for the activity of restriction enzymes at a single point, because wherever you cut in that place only you have to insert the, see, this part should be inserted here, means the sequence should match, right? The sequence should match, means the same restriction endonuclease you have to use here and the same restriction endonuclease you should use here. So it cuts at the same place and it cuts in the same place so that this sequence can be added here, right? So this plasmid or this vector should be capable to undergo the cutting, right? If it is not undergoing the cutting, then there's no use, right? So it must have a unique cleavage site, okay, for the activity of the restriction enzymes. So it should have the same DNA sequence, just like in the, I mean, the way you're cutting the, that particular desired DNA segment, right? So it must be able to replicate truly inside a host cell and its introduction. Okay, okay. So after introducing the foreign DNA into this vector also, then it should replicate. It should not die, right? It should replicate. Then it must contain genes which produce, I mean, which provide resistance to antibiotics. This helps in the selection of transformed cells from untransformed cells. Okay, so it should be resistant to the antibiotics also, because we use different types of antibiotics not to cause any antimicrobial contamination, sorry, microbial contamination. Okay, that anyway, when you usually work in the laboratories, then based on experiences, you will get a lot of things. But For examination purpose, if you just mention like one or two, that's enough. Then these are the vectors. Just now I told you plasmids, bacteriophages, cosmids, artificial chromosome vectors, and expression vectors. Okay. Now coming to the vectors. Different cloning vectors are available. See, for example, E. coli, phase lambda, caspid lambda, then plasmid, artificial chromosome, PSC, then bacterial artificial chromosome, yeast chromosome. So these are different chromosomes. But in this one, you can insert up to 5 to 25 kb of the foreign DNA. Got it? See, uh, this is the plasmid. Let us assume this is a plasmid. In this, you can insert up to this one only, right? So if you can ligate like this, I mean, if this endonuclease is cutting this area, so you can insert more foreign DNA, right? So how much they allow the foreign DNA means here E. coli lambda, I mean phase lambda, okay, that will allow 5 to 25 kb only. We can insert the foreign DNA only 5 to 25 kb. Then cosmid 35 to 45. So based on requirement, if you want to insert more DNA, then you choose the different vector. Okay, yeast Saccharomyces cerevisiae. So that can accept 200 to 2000 kilobytes of the nucleotide strand. That is a foreign DNA, right? So it will accept. So like this, we have different types of vectors, right? Now, okay, let us talk about the plasmid. So here I made the note of different vectors, right? Then coming to the plasmids. So what's the plasmid? The plasmid is the extra chromosomal material, as I told you. Okay. So here in one plasmid, you can add two or three, uh, foreign DNA also. But if you're talking about one plasmid, this is a PUC19. Okay. So it's from University of California and researchers designed as 19, PUC 19. Okay. UC means University of California. PUC means Plasmid University of California. So they named it as 19. Initially, they extracted this one, it seems. Okay. So this is one PUC 19 plasmid. Okay. So these are the characteristics. So it is having a lag Z, then hint 3, BOM H1, echo RA means these are the sites where the endo, I mean, restriction endonucleases will work to cut this one. Okay, like this, we have different sites. Clear? Now, larger DNA fragments in cosmids and other vectors. See, this is the cosmid vector. Cosmid vector. It can accept multiple, DNS. Multiple DNS means foreign DNS. 35 to 45 kb also they can accept. Okay. Then from E. coli, you are extracting an enzyme. You're extracting an enzyme, and later, sorry, you're extracting a plasmid. Okay, or we can consider this as a bacteriophase. See, what's a cosmid? Cosmid is nothing but a bacteria is infected with a bacteriophage, right? So this bacteriophage inserts its DNA into the bacteria. So this is called the cosmid. Okay. So in this cosmid, we are ligating or we are we are doing the recombination of a big DNA sequence. Okay. By cutting these DNA sequences, these sequences will inserted here, and later they may enter into different, bacteriophage again, because here the bacteriophage will enter here. Then once the replication finished, the bacteriophage only will get released. Right? These are the recombinant cosmid vectors. Later the expression will be collected. Clear? I hope you know about this bacteriophages. I think you might have studied in maybe intermediate. I'm not sure. But bacteriophage, lytic phase and lysogenic phase. There are two phases in the bacteriophage development. Okay, now coming to the host. Okay, I hope you understood about the the cosmids and plasmids. Then coming to the host. Host means after recombination, you can keep it in the prokaryotic cell or eukaryotic cell. See, take the bacteria, which is a prokaryotic, you can insert by methods called the transfection, transduction, conjugation. You can use different methods here. and insert it into a bacteria. What's the bacteria? Maybe E. coli, Bacillus subtilis, and Streptomyces species. If it's a eukaryotes, then you can insert it into the fungi. Which one you are going to insert? Recombinant DNA. Fungi or animals, and in the plants also you can insert. Okay, you can infect, right? Now, production of recombinant DNA using a human gene and bacterial plasmid. The same thing I explained you. Okay, see, this is a human chromosome. So this is a part where the insulin is getting secreted. That is the insulin gene. Cut with the restriction enzymes. Take the bacterial plasmid. So this is the bacterial plasmid. Okay. Then you, after cutting this, okay, this red part, and this is the plasmid. You cut the plasmid and remove this part. There is a space. Okay. Then you make it linear, make it straight line. Okay. Then in this you add this part. Then it will become a recombinant DNA. You combine this with the DNA ligase. Then after this, you keep into a cell or host. Then it will get doubled, right? See, it will get multiplied, right? Like this, one become two, like this. Then after this, what it do? Because of this gene, it will express the insulin. Then collect the insulin, and later you can use it for the patients who are having the deficit of insulin, right? So this is the same thing. Then what are the steps here? First, you should cut the isolated, we should isolate the DNA, the desired DNA. So what are the methods of isolating the DNA? Means we have different methods. Okay. First, you should lyse the cells because the DNA present in the cells, right? You have to rupture the cells. Okay. When you use the detergent, it will generally rupture the plasma membrane of the detergent. Later, cell contents will be there. You add a membrane called the protease, then the entire protein content will get dissolved. Then there won't be any protein, right? Right? Then you add a RNase. What is RNase? RNase means it will completely destroy the RNA. Then add the RNase so that it will destroy the RNA also. Means we are removing the cell membrane so that the entire cell content will come out, and later you are denaturing the protein as well as you are denaturing the RNA. Then what is present here? It contains the cell debris and the DNA only, right? Because we are using a protease to destroy the protein and RNase to destroy the RNA. The remaining will be the complete cell debris and the DNA, right? Then centrifuge this one. After centrifuging, the supernatant contains the DNA. This is the yellow color, right? The supernatant came. So that is the DNA. You transfer it to some other test tube by removing this cell debris. Now the test tube contains only the DNA part, right? Then you precipitate with ethanol, add ethanol, it will get space. It will, it will become like a thread-like structure. That's the DNA. Then spool it using a glass rod. That's the DNA. So this is how you have to extract the DNA. Got it? I mean, there are different methods to extract the DNA, right? But practically, I mean, based on the requirement, then we'll be using different types of proteases and rhinases, okay, RNases, right? Okay, then after extracting the or isolating the DNA, then the DNA be cut into large fragments by mechanical shearing. Mechanical shearing means we have different methods. Put the glass beads, okay, and shake it. The glass beads will cut the DNA in different as a different small parts. Okay, that is one irregular cutting. Or you can use the restriction endonuclease so that it will cut at the selected places only. Okay, like that you cut the DNA. See, you're cutting this DNA with a restriction endonuclease so that it becomes a sticky ends like this. Then see this example. This is a DNA part. Okay, you're cutting this one. Sticky ends came. See, these are the sticky ends. Okay, then remove this part. Only you just take this one. And later from the plasmid, also use the same enzyme. So in the plasmid also it will cut this part only. Then remove this part. Okay, remove this part. Then later take this from the plasmid and add this desired in here. Okay, it will get attached by ligase. Then circled. So this is the recombinant DNA, joining the DNA. Then after that, after the recombinant DNA, then you have to search for a host. Okay, how it should enter into the host cell, but will the host cell will allow the foreign DNA? No. So the cells are treated with calcium chloride. Okay, the cells are treated with calcium chloride. Then DNA is added. Cells are heat shocked at 42 degrees centigrade. then the plasma membrane becomes semi-permeable, then it will allow the DNA into the cell. DNA goes into the cell. Once in a cell, the recombinant DNA will be replicated. When the cells divide, the replicated recombinant molecules go to both the daughter cells, which themselves will divide later. Thus, the DNA is amplified like this. So, we are using different enzymes here, and we are using CaCl2 and shock also 42 degrees. Okay, later in 20 cycles, you will get these many copies. Okay, the replication will be faster. These many copies you will get. Okay, this is called the amplification. See, this is one more slide. Generation of DNA fragments. Restriction enzymes. Okay, digest the DNA or cut the DNA. Okay, and later insert into a cloning vector, then ligate it. After that, introduction to the host cells. How to insert into host cell? By transformation, transfection, transduction, or conjugation also you can use. And later, once done, you do the hybridation, PCR techniques or other methods, okay, to purify them. Okay, like this. Now, gene cloning strategies. Just now I told you, right? We have different methods: transformation, transfection, transformation, conjugation, electroporation, then liposome mediated gene transfer, direct transfer of DNA or DAN. Okay, then gene libraries, screening methods. Okay, like this. Later, what are the techniques used in rDNA technology? rDNA technology, we use different techniques: gel electrophoresis, cloning libraries, PCR technique, restriction enzyme mapping, nucleic acid hybridization, DNA microarrays. We use different techniques in the rDNA. Why these techniques? See, for example, you want to test whether this particular restriction endonuclease is cutting that particular part only. Then there is a restriction enzyme mapping. That test we have to do. Then uh, to know the molecular weight, then we use the gel electrophoresis. Okay, like this we have different techniques. See, based on requirement, then we'll be using different techniques. Okay, then way to use this rDNA technology. So this rDNA technology means those recombinant DNA products. We can use it in the agriculture, pharmacology, like artificial insulin production. Just now we discussed drug delivery to target sites. The medicine like gene therapy, antiviral therapy, vaccination, synthesizing clotting factors, fluorescent fishes, glowing plants. Then in the drug discovery also we use this rDNA technology. So hormones, cytokines, interferons, the antiviral, interleukins, enzymes. For vaccinations also we use this rDNA technology. First, human insulin only we discussed, right? So the rDNA technology made human insulin is available currently in the market, okay, which will treat the type 1 diabetes, right? So they are available with different names like what is it? Humu2N or Humuin, then Novolin. Okay, they are available with different brand names. Okay, see the same thing I explained. Then DNA vaccines also available in the market. Or DNA vaccines like hepatitis B, okay, HCV. A lot of DNA vaccines are available in the market. Then interferons also, antibiotics, and therapeutic human proteins also they are producing by the rDNA technology. Anemia, hemoglobin erythropoietin. So these are produced by recombinant process only. Asthma, interleukin 1 receptor, obturation, relaxin, blood clot, tissue plasminogen activator, and urokinase. These help in the clotting mechanism. Burns, epidermal growth factor. Means if a person is lacking the blood coagulation factors, then they need these enzymes, tissue plasminogen activator. Okay, so these are also prepared by the rDNA technology only. The diabetes, emphysema, female infertility. Like we can create the HCG also. Artificially, we can create the HCG also. Human gonadotropin for female infertility. Then superoxide dismutase, free radical damage to damage the free radicals. For heart attack, you know, pro urokinase, hemophilia A, factor 8 we can create. Hemophilia B, factor 9. So after this rDNA technology, we are creating lot of artificial proteins. Okay. Even not only so vaccines also we are developing. Okay. And some are still they are being developed like rotavirus, chickenpox, encephalitis, genital ulcers, liver damage. Okay. Upper and lower respiratory tract lesions, dysentery, gonorrhea. A lot of you know vaccines we are developing using this technology. Like this is called the rDNA technology. Clear? So I hope you understood the rDNA technology. Simple. rDNA technology means just keep the desired gene in a plasmid or any extra chromosomal content and place it in one organism. That organism will multiply and it will give the desired product of that particular desired gene. That's it. Then you have to extract them or you have to separate them and you can use it. Okay. So this is the rDNA technology. Clear? Okay. Now there is one thing called hybridoma technology. So we'll be discussing this hybridoma technology also. Hybridoma technology is nothing but the rDNA technology, but in hybridoma technology, we are using the OMA. What do you mean OMA? OMA means cancer, right? Carcinoma, sarcoma, lymphoma, right? OMAs. OMA is nothing but a cancer. Okay, OMA. Based on the area of the cancer, okay, we call it a OMA, right? Sarcoma. If it affects the lymphocytes, it's a lymphoma. Okay. Mm, if it affects the myelocytes, myeloma, chronic myeloid leukemia, CML, ALL, acute lymphoid leukemia. Okay. I mean, we call it a OMA. OMA is a cancer, right? So hybridoma technology also, it is related to the rDNA only. Okay, so this rDNA technology developed in 1970s and 80s, but this hybridoma technology developed in 1976. Okay, 75 or 76. Okay, and they got a Nobel Prize also for this in 1984. So hybridoma, okay, I will explain this hybridoma in detail. I think this is okay, fine. There is one more slide. Okay, you can, for examination purpose, you can use this part. This is enough. Okay, but I will explain in detail this hybridoma technology. I have one more slide with me. Yeah. So monoclonal antibodies, preparation of monoclonal antibodies using hybridoma technology. Okay. So using this hybridoma technology, we are creating monoclonal antibodies. What do you mean a clone? Clone is nothing but the same. Clone is nothing but we are creating the same DNA. We are replicating the same type of the DNA. That is nothing but the clone, right? So using this hybridoma technology, we are creating monoclonal antibodies. So what is the purpose of antibodies? Tell me, what's the purpose of antibodies? What the antibodies do? What the antibodies do? Any idea?
Wo-
Yeah.
Works
Def
against antigen.
yes, they work against the antigen, right? So what do you mean by monoclonal antibody? What do you mean by monoclonal antibody? Means antibodies are coming from a single clone. So the antibodies will be the same, right? See, by example, from childhood onwards, we prone to different diseases, right? See, for example, I got typhoid. Then usually typhoid antibodies will be there in my body, right? Then I took multiple vaccines, like hepatitis vaccine I have taken. Then HPV vaccine also I had taken, human papilloma vaccine also I had taken. I have taken tuberculosis vaccine. Okay, in my this 50 plus years of age, I infected with multiple, cold, and I mean the bacteria and maybe influenza and other viral diseases also. So my body is having a lot of antibodies, right? So if you collect the serum and you separate the antibodies, then you can find antibodies of multiple origins, right? Multiple types of antibodies you can see. So those are all polyclonal. Polyclone means one clone will give you. So for example, I'm like, for example, I'm exposed to a typhoid. Then my body will create the antibodies against the typhoid. Means one B cell, okay, it will liberate the antibodies. So when my body is having typhoid antibodies now, but in my blood, already a lot of antibodies will be there, right? So those antibodies will develop from different clones, right? So that is a polyclonal antibody. Monoclonal antibody means it contains only one origin, means one type of antibodies only, because all these antibodies are developing from one clone only. Got it? So, for example, uh, I infected, I infected a mouse or a mice or any animal, okay, animal with some disease. For example, I injected some, a bacterial disease, okay, a bacteria I injected into a mice, let us assume. So in the spleen, the mice B cell will get responded to this disease, then it will release the antibodies, right? Then I will pick that B cell which is releasing the antibodies, right? I will pick that B cell. In the B cell there is certain region is there which is liberating the antibodies, right? Then I will extract that region. Okay, after extracting that region, I will keep it into a plasmid and then I will transfer it into an organism. Okay, then that organism will start producing the antibodies, right? So those antibodies again is that particular disease only, that one particular disease only, right? So that is nothing but the monoclonal antibody. Then you collect the antibodies, okay? Then those antibodies again work against to one particular disease only. So it contains no any uh, other antibodies, right? So it is a purely these antibodies are specific to one disease only because these are originating from a single clone, right? So that is the reason we call this as a monoclonal antibody. Okay, these are all the monoclonal antibodies. They work perfectly against a disease because there is no any contamination with the other antibodies, right? No other antibodies are there here in this. It's purely related to one clone only, right? So that is how we are creating the monoclonal antibody. But here, the antibody production will take much time, right? So that is the reason what we are doing, we are adding a cancer cell. We are adding a cancer cell. Hybrid OMA means we are adding a cancer cell. So what happens if we add a cancer cell? The simple logic. One cell is one cell is producing the antibodies. Okay. So one cell should divide into two cells, right? And there is one mitosis, meiosis stages will be there, right? So to divide one cell into two cells, it will take a lot of time and also there is a regulation. Okay, there is a some factors which will stop the growing of cells rapidly. So that is the reason what we do. We add the oncogenes also. Oncogene means cancer causing genes also we add to the cell. Okay, now the cell is having a particular desired gene as well as a cancer gene. Then what happens here? Hybridoma means you are hybriding or you are hybridizing your desired gene with a cancer gene. Then it will get both the characters, right? What the cancer do? It will replicate or it will divide or it will multiply rapidly, right? So one will become two, two become four, four, like this. It will multiply very fastly. Okay. And when the cells are multiplying fastly and they have the ability to release this monoclonal antibodies, right? Then they will be releasing the monoclonal antibodies. Means the cell division will be faster and each cell is liberating the or each cell is expressing the antibodies. So the huge production of antibodies or monoclonal antibodies will be there when you infect a cell with a cancer cell. That is nothing but the hybridoma technology. Clear? Okay, I'll show you here once again. Monoclonal antibodies. Antibodies are produced by a specialized group of cells called the B lymphocytes. Usually antibodies are produced by B lymphocytes only, right? When a foreign antigen, when a foreign antigen enters the body, enters the body, due to immune response, B lymphocytes develop into plasma cells, liberate antibodies or immunoglobulin like this, IgE, IgD, different antibodies they will liberate. Then what is the monoclonal antibody? Just now I told you. Monoclonal antibodies are antibodies that are identical because they are produced by one type of immune cell, all clones of a single parent cell, right? What is a polyclonal? So it is having multiple clones. Okay, it's having the product of multiple clones, like multiple antibody types we have. That is the polyclonal. Okay, then polyclonal antibodies means clones of many B cells. Okay, it is having multiple types of antibodies. Okay, so if there is a cell, so these antibodies will attack to all because it's a target. Target is having multiple antigen binding sites. So these antibodies, multiple antibodies are there. Each antibody will bind to that antigen, antibody binding site. Okay, these are non-specific. But here monoclonal antibodies means specifically it will bind to only one site. Okay, one epitope only. So monoclonal antibodies are identical immunoglobulin generated from a single B cell. Okay, these antibodies recognize unique epitopes or binding sites. Okay, this is fine. So we have different types of monoclonal antibodies like murine monoclonal antibodies, chimeric monoclonal, humanized monoclonal, then human monoclonal antibodies. Like this, we have different types based on the origin. See, you are creating a monoclonal antibody from a human, then humanized, I mean human monoclonal antibodies directly. Humanized means you are provoking, okay, and getting them, but not exactly from the human beings. Okay, so you're using some other animals also. Then chimeric means you are combining with again a different DNA. Then murine, completely mouse. Okay, see, it's a murine, mouse, chimeric, having both. Then humanized, having the others also, but mostly the human. Then the human means completely human origin. Okay, see this one. Human derived 100%. It's completely, sorry, mouse derived 100%. Murine means these antibodies are derived completely from the mouse. Then these are the, see, these are available, right? See, for example, uh, mm, see, HIV patient is there, or human immunovirus attacked patient, like AIDS patient is there. So he cannot synthesize the antibodies on his own because of the disease, HIV disease, because his body is not capable of producing any antibodies because of the immune suppression, right? So he got infected with some disease, let us assume. His body cannot develop the antibodies, right? So what should we do? In the market, lot of artificial antibodies are available, right? So if he's got attacked with maybe diphtheria, then diphtheria antibodies are available. So when you are purchasing these antibodies, then there is a label on the antibody. These are murine origin or murine antibodies. Means these antibodies are completely developed from the mouse. Means they give a diphtheria toxin to the mouse, then the mouse will release the antibodies. Then they will extract the mouse B cell, and they will produce the antibodies from that particular clone, and they will prepare artificially. They will sell as a murine antibodies, right? So that clone of antibodies is the origin of the murine, right? Then 33% means chimeric antibody. Chimeric antibody means some antibodies are human originated and some are from the mouse. Okay. Then 100% humanized means like this. It's not 100%, 10%. Means only little bit the other animal, then remaining is the 90% is the human. If it is a completely human origin means that is 100%. Okay. Then advantage of monoclonal antibodies is specificity. It will, specifically attach, and it is more expensive. Okay. Then what is the use of monoclonal antibody production? It is for immunization purpose, cell fusion purpose, selection of hybridoma, screening, and then cloning and propagation. First, what they will do, they will immunize. Okay, I will show a diagram so you will understand. See, there is a mouse. The mouse is immunized with antigen X. Okay, let us assume tetanus. Tetanus. Okay, so here the mouse is given the tetanus antigen, and the mouse spleen produces plasma cells that secrete antibodies against the antigen. Right. So let us assume these are the plasma cells. The plasma cells will liberate the antibodies against this particular antigen called the diphtheria. Okay, diphtheria you take anyone. Okay, now these cells are capable of releasing the antibodies, right? So what these are? These are antigens we injected. So these are the antibodies. The mouse spleen is liberating, right? Then after collecting all these, which one? The antibody producing plasma cells, you collect them and place it in one media. Okay. Then later there are some cells are there. These are the cancer cells or myeloma cells. Okay. So select some myeloma cells. Okay. Then after selecting these myeloma cells, add these myeloma cells here. Now in this we have the cancer cells as well as the uh, spleen, I mean the plasma cells extracted from the mouse. Then keep these in a medium. Keep this in a medium. Medium is for the growth, right? So keep this in a medium. And make artificially that the fusion should happen between these cells. The fusion should happen. Okay. So there is a fusion between these cells, myeloid cells as well as the spleen, I mean the plasma cells, right? Now, but the fusion may happen in different ways. See, sometimes both the plasma cells may combine. Plasma cells may combine. So, but when the both the plasma cells are combined, then there won't be any, myeloma cells activities comes here, right? So there are four chances are there, right? What are the four chances here? There is a chance of combining two myeloma cells. That's not needed. Then there is a chance of combining two plasma cells. Okay, that may happen. See, cells are transferred to hat medium. Okay, so here there is no any combination or there is no any fusion. The plasma cell I mean this plasma cell, right? No, here the fusion happened. See, myeloma cell and plasma cell, here the fusion happened. This we need. This we need. But unfused plasma cell. So there is a plasma cell which is not fused. It may die in the hat medium. Okay. Now, uh, some are unfused. Okay. Unfused myeloma cells also they die. Or fused myeloma cells also they won't die. Only we have to extract the cell which is fused. Fused means the plasma cells and the myeloma cell fused cells only we have to extract. Okay. And then grow separately. Then this contains both the characteristics of the cancer cell as well as the producing these antibodies. Right. Now, uh, grow them, then it will yield the product. What that product contains? That contains antibodies against the that particular disease. And here the growth also will be fast. Clear? Okay. See this one? Immunization. Then isolate the B cells. Then these are the myeloma cells. Fuse them. Okay. Then select them using a HAT medium. Okay. Then screen them. What are all the fused cells? After screening, you extract them. Then it will yield the monoclonal antibodies. Clear? Again, the same steps, immunization, cell fusion, like this. Okay. And what are the applications here? The monoclonal antibodies. MBS means monoclonal antibodies. Therapeutic you can use for protein purification, for diagnostic applications also we can use. So these are the different advantages. Okay. Just go through this, you will understand. There's a big list also. The use of antibody, abciximab. Okay, it's a chimeric. It's for cardiovascular disease, inhibition of glycoprotein 2A and 2B. Okay, so bevacizumab, Avastin, brand name Avastin. It's a humanized vascular endothelial growth factor to reduce the colorectal cancer, age-related macular degeneration. Avastin. Like this, we have different, monoclonal antibodies are available in the market. Yeah. So this is about the monoclonal antibodies. Clear? Yeah, that's it for today. Yeah, just discuss among yourselves and let me know. Anyway, tomorrow I think we don't have the class. Tomorrow class will be taken by some other faculty, DRP. Okay, she'll